Hello, I am the Missile Guidance System, and I'm back again to teach you about... Oh, this is going to be a long one. I don't get paid enough for this. So we're going to start from the beginning. No, not that beginning. I mean with the first standard, Bio 1.1. We have 3 objectives in this one. Let's get through them fairly quickly. Organelles. As do most multicellular organisms, many cells also have structures that allow them to complete functions and maintain homeostasis. Okay, let's speed run the different organelles. The ribosome is the factory for proteins. The mitochondria is the mighty powerhouse. The nucleus is the brains of the operations. The lysosomes is the Lysol. The cytoplasm is the open seas. The vacuole is the storage. The cell membrane is the guard blocking the entrance to the club. The chloroplast doesn't think the sun is a deadly laser and embraces it. And the cell wall is just a brick wall. Anyways, onto the types of cells. There are 2 general types, eukaryotic and prokaryotic. The biggest difference is, one has a brain, the other one doesn't. I mean, just look at their IQ scores. See, the eukaryotic one has a nucleus, while the prokaryotic cell does not. In fact, prokaryotic cells don't have membrane-bound organelles, which is pretty much most organelles. They only have ribosomes, DNA, cytoplasm, and a cell membrane. Usually they are bacteria. Now we have cell differentiation, which is when unspecialized cells called stem cells have certain genes activated, which will allow them to evolve into a specialized cell like a muscle cell. Bam, boom, bop, done with it just like that. Now we're on to the next standard. This standard has a few objectives, but I will only be doing one, the cell cycle. This one is a bit complicated. In total, there are 2 phases with multiple subphases. These 2 phases are interphase and cell division. In interphase, the cell is growing and replicates its DNA. The subphases of interphase are G1, S, and G2. In G1 and G2, the cell is growing, while in the S phase, the DNA is being replicated. Now we move to cell division. This is where things get a little bit confusing. So in cell division, there are 2 subphases, mitosis and cytokinesis. In cytokinesis, the cytoplasm divides, and in mitosis, well, there are subphases in the subphase. To be exact, there are 4 phases of mitosis: prophase, metaphase, anaphase, and telophase. In prophase, chromosomes become visible. In metaphase, the chromosomes are lined up in the middle. In anaphase, the chromosomes are pulled apart to the poles by spindles. Then, in telophase, the chromosomes on each pole are encircled by a new nucleus. Now, before we finish this topic, there is one more thing: meiosis. Meiosis is pretty much mitosis with very important differences. In meiosis, the mitosis process occurs twice. Crossing over occurs allowing for genetic variation. And by the end of it, you will end up with half the amount of chromosomes in each cell compared to mitosis. Also in mitosis, you will have 2 cells at the end. But in meiosis, you will have 4. This process only occurs in reproductive cells. Okay, so that's finally done. On to the next. Time for DNA and RNA. So DNA and RNA consists of the macromolecule, nucleotides. A nucleotide consists of 3 things: a sugar molecule, a phosphate group, and a nitrogenous base. These nucleotides form a ladder. However, a strand isn't considered this. It is actually considered this. This is a single strand of DNA. And believe it or not, There are 4 different kinds of nitrogenous bases: adenine, cytosine, guanine, and thymine. Or as you simpletons would put it, A, C, G, and T. Each one of these bases has a complementary base. For example, A with T, C with G. Ah, like true love. So this means when you have a pair of DNA, the pairs will be complementary to each other. But now, what about RNA? Now, RNA is a little different. For one, you will never see RNA in a double helix. And RNA does not have thymine. It is replaced with uracil, aka U. Now, you might be wondering, how are these letters important? Well, your body uses them as code for proteins. Yes, you heard that right. DNA is what codes you. This is just a simulation. You are just a coded being. Anyway, as I was saying, the basis of DNA is what the mRNA uses to tell what the tRNA needs to bring to the rRNA or ribosomes to create a protein through a sequence of amino acids. The ribosome reads sets of 3 bases in the mRNA. A set of 3 bases is called a codon. But now what about mutations? Mutations are quite simple. When you have the DNA, a substitution, deletion, insertion. Substitution is when a base gets swapped out. Deletion is when a base is removed. And insertion is when a base is added. Mutations can either be neutral, harmful, or helpful. Now on to genetics. Remember this? This is important. You should remember how to do Punnett squares because I am lazy and don't want to explain them. But I will explain incomplete dominance, codominance, and multiple alleles. Incomplete dominance is when the middle ground of 2 traits are shown because the dominant allele does not show through completely, while codominance is when both traits show up. And multiple alleles is when a trait is determined by multiple alleles instead of 2. Would you look at that? We're almost done. Man, I think I need a quick break. This reminds me of something. Wait, I remember. It's like evolution. Similar to how companies will upgrade their products to fit the needs of their customers, populations of organisms will have mutations that help them in their environment. Like iPhones, the electronics may be similar, however the function is different. Same goes for organisms. If a structure is similar yet functions differently, this is called a homologous structure. And if the structure is different yet functions similar, this is called an analogous structure. There is a mechanism of evolution called natural selection. This is where, in the population of organisms, an organism may have a mutation at random that allows them to easily survive and reproduce. There are 3 forms: directional, stabilizing, and disruptive. Directional is when one trait is favorable in the environment. Stabilizing is when the middle ground of 2 traits is favorable. And disruptive is when 2 traits are favorable. Oh yeah, I almost forgot. Speciation is when 2 species are isolated in some way, causing them to evolve into different species over a long period of time. Just like how my ex isolated herself away from me. Anyway, have you heard of biotechnology? Well, in a nutshell, it's when DNA is cut by an enzyme and must go through a race on a gel plane. The biggest pieces will be the slowest and be closest to the beginning, while the smallest pieces will be the fastest and will be farthest from the beginning. This is called a DNA footprint, and it can be used in crime scenes and paternity. What's that? You forgot what an enzyme is? Well, it's a protein that speeds up reactions by doing this. Got it? Good. Wait, that's it? We're finally done. Well, I didn't go through every single topic. However, I hope this little summary helps with the main topics. Now it's time for me to get my paycheck. The missile knows what it wants at all times. It knows this by adding work and energy, which will equal to money.